Good afternoon, everyone. It's 12 noon, so we'll get started with our afternoon presentation. Before we start with the official presentation of medical grand rounds today, Dr. Mark Roger has asked for a few minutes to speak to members of the Department of Medicine. So, Mark, if you want to take over, and then we'll move on to the grand round presentation. Thank you.
Great. Thanks, folks. I'm assuming you can see my slides. All good? Great. Thanks. I just wanted to provide a bit of an update on our strategic planning exercise for the McGill Department of Medicine. Both an update, but also an invitation as well. As you know, we embarked on this participatory community building blank page forward-looking exercise last fall. And I assume that we've been in your inbox enough times that you have a good sense of what we're trying to do. We're starting out with defining our aspirations, and we've just completed that phase. And we're moving into the tools and processes phase. So how do we reach, what do we need to put in place? What do we need to have in order to achieve our aspirations? And the last phase will be defining metrics. What do we measure along the way to know whether we're there or we're getting there? So we started in the fall with the aspirations phase, harvesting aspirations from the community, theming the aspirations that were harvested. Then the community ranked the aspirations. So in terms of harvesting the aspirations, over 120 members provided us with 180 aspirations. And we themed them into 7 aspirational themes. A lot of the 180 overlapped. Then we asked the community to rank the themes, the 7 themes. And these are the 7 themes, and these are the aspirations that fell within the 7 themes. And after the ranking exercise, we whittled them down to 4 themes. I love working here, effective advocates for the best patient care and resource-constrained environments, the training center of choice, and the most research-intensive department in the country. Next, we took all of the aspirations, 7 aspirations in each of the themes, and got you to rank the top 3. And I'm showing this particular data to show you that we had broad representation across career paths, age groups, sites, really in all of these aspiration ranking exercises. So our top 3 aspirations for the training center of choice, I'll let you absorb them. But number 1 was that our core IM programs attract the best candidates and provide the best core IM training in the country. Number 2, that we provide a healthy and safe learning and working environment at all levels. And number 3, our clinician teachers are valued, respected, and master clinician teachers are celebrated. So those are the top 3 for the training center of choice. The I Love Working Here had 8 aspirations. Again, good representation of folks ranking the aspirations. And the top 3 I Love Working Here aspirations were, number 1, the place where digital and other innovations lead to top-notch provider experience and administrative excellence. 2, where leaders and colleagues and institutions care about me as a person and value and respect my contributions. 3, that our leaders and faculty members are as diverse as those we serve. Next, for effective advocates for patient care, the top 3 were developing and delivering digital innovations that improve access to quality of care. So state-of-the-art EMRs, patient-facing apps, virtual care options, contributing and advocating for solving the current and coming health human resource crisis. And 3, rendering state-of-the-art diagnostics and therapeutics accessible to all Quebecers. And then the last theme was the most research-intensive department in the country. The top 3 aspirations were to grow the number of clinician scientists we have in the department and ensure that they provide the they provide and produce the most impactful research in Canada. 2, that we have PhD scientists and clinicians co-creating in the most translational department of medicine in Canada. And 3, that we have more world-class research teams than any other department in Canada, teams being multidisciplinary scientists working in a specific area. So those are the top 12 aspirations for the department's strategic plan. Now we're moving on to what we need to put in place and what tools need to be there in order to deliver on these aspirations. So over the next month, and this is the invitation part, we've got 4 town halls, which are essentially small group breakout session brainstorming sessions. And we'd like to invite you to follow your passion. If you're passionate about one of these themes, please come. You're more than welcome to come to all 4 teams. We know it's a bit of a theme meetings. We know it's a bit of a time commitment, but please come to at least one. And then after we do this, the executive will work on the metrics, and we're going to write the final product in the summer. So thanks for your attention. I'll turn things back to Dr. Giannetti and friends, and look forward to hearing from Dr. Sebag.
Thank you, Mark. Thank you for this initiative. Very interesting, actually. Thank you so much for sharing that with us. We're going to move on now to our medical grand rounds presentation. Before the presentation begins, I would just like to make a brief land acknowledgement. McGill University is on land which has served as a site of meeting and exchange amongst Indigenous peoples. We acknowledge and thank the diverse Indigenous peoples whose presence marks this territory on which peoples of the world now gather. I'd like to ask you to please remember to enter your questions in the chat at the end of the presentation, and please answer a brief survey that will be sent to you right after the grand round presentations. And now, without further ado, I'd like the Chief of the Division of Hematology, Dr. Chantal Séguin, to introduce our speaker today. Dr. Séguin. You are still muted. Chantal.
Thank you, Dr. Giannetti. It's a welcome, everybody. Good afternoon. It's a pleasure for me to introduce Dr. Michael Sebag. I have known Michael very well over the last year, since at least, I would say 2008, if not more. Michael is one of our hematologists in our group. He initially studied at McGill University, completed a bachelor degree in science in the field of microbiology and immunology. And then he completed a PhD degree in 1996 in the field of physiology, moved to Toronto to perform a medical degree in medicine that he completed in 2000, and then came back to McGill University and completed an internal medicine and then an hematology fellowship at McGill University, and then moved to Mayo Clinic to perform a myeloma fellowship there, and was recruited as an assistant professor in 2008 here at McGill, initially at the Montreal General Hospital, where we've been working together since then. He became associate professor of medicine in 2020. He's been holding many scientific director positions, such as scientific officer for the Myeloma Canada Research Network that he's involved in since 2017. He's also the director of the McGill Multiple Myeloma Research Group since 2010. And since 2021 has been the medical director of the McGill Immune Effector Cell Program. He's been working extremely hard to have the CAR T cell program organized and developed at McGill. We were very proud of his achievements. And also he has, of course, published multiple publications in excellent journals in the field of myeloma. He holds currently a few CIHR grants as a co-PI, and also a current grant as an investigator-initiated trial on a specific clinical trial involving some some molecules involved in multiple myeloma. So it is a great honor for me to present Michael Sebag this morning, and I'm inviting him to start his talk on the overview of the CAR T-cell program at the MUHC.
Thank you, Chantal, and thank you, Mark. Thank you, Chantal, for the very kind words. Let me try and get this. Okay. Let's see if this works. There we go. Okay. I hope everybody sees a full screen. Perfect. Okay. Thanks, Mark. So we'll talk about CAR T, or chimeric antigen receptor T-cell therapies. I'm going to talk about the history. I think we've done that before. This talk is just to reacquaint yourselves about concerning the indications, the clinical indications, and then what we've been doing at the MUHC, how we structured it, and how we went about starting such a program. So for those who don't know, a chimeric antigen receptor T-cell therapy, oh, I don't know if somebody could be muted. I'll try. There we go. There we go. So a CAR T-cell is essentially a T-cell, an autologous T-cell, so a patient T-cell, where we've co-opted its function by genetically reprogramming it. So using lentiviruses, we put in a construct that'll code and then translate for a protein that'll insert itself in the membrane of the T cell. And that protein will have a transmembrane domain. It'll have an antigen binding site, and that's the antigen binding site of an immunoglobulin, and an immunoglobulin that has been selected specifically to target something at the surface of hopefully a cancer cell. And distal to that, within the intracellular domain, second generation CAR T cell products have both a co-stimulatory domain and a T cell activation domain. And so the co-stimulatory domain in particular is very tonically active. This gets the cell in a very active state, ready to go, ready to fire. And the T cell, no matter what it was destined to do, once we plucked it out of the patient, it's now reprogrammed to hit only one target, one of our choosing. So the process is as such, it's not terribly complicated, but it's nonetheless still quite the endeavor. It starts with a patient, obviously, who undergoes a leukapheresis, a little bit like a dialysis machine, where we spin out the peripheral blood mononuclear cells, all peripheral blood mononuclear cells. And then we, with the help of antibody-coated beads, these are then selected, the cells that we want. So the CD3 cells, rather, the T cells are then selected, and the others are washed away. And then those CD3 positive T cells are then transduced, usually using a lentivirus or retrovirus, and to express the construct. They're then expanded in culture. The bead is removed. The product is then frozen and then returned to the patient. And the patient receives a low dose of chemotherapy called leukodepletion. And that really just quells the immune system so that they don't immediately reject the cells. The cells are autologous for the most part, but nonetheless, they've been manipulated. They have a new antigen at the surface, so there is a possibility they could be rejected. And so this, basically abrogates the immediate rejection. The process here is done, we have a choice of either doing it in-house, if an academic CAR T, or done in a pharmaceutical company as for a commercial CAR T process. We'll be speaking largely about the commercial CAR T. So what happens once the cells enter the circulation of the patient? So the CAR T cells will find their way to the tumor. They'll bind by virtue of this chimeric antigen receptor. And that binding will, kill tumor cells, but also release neoantigens from the interior of the tumor cells, and also release cytokines, which will recruit other inflammatory cells, including other T cells that are in the patient, as well as monocytes, for example, macrophages, whatever are in the area. So that recruitment assists in the cell killing. So we'll talk briefly about the indications. And the first indication is lymphoma in diffuse large B-cell lymphoma. And one of the pivotal trials is the Juliet trial. So a trial using a product called Kymriah or Tisa-cel. And in that trial, these were CD19-directed CAR T cells with a costimulatory domain called 4-1BB. And in this trial, a phase 2 trial, about 100 patients were enrolled with refractory relapsed diffuse large B-cell lymphoma, or transformed follicular lymphoma, or high-grade B-cell lymphoma after 2 lines of therapy. So these are patients that had at least 2 lines of previous therapy, many of them autologous stem cell transplantation. And about half of them, in fact, had an autologous stem cell transplantation. So this is not upfront. This is for patients who really have relapsed and have few recourses. And in this trial with this CD19 CAR T, the overall response rate was 52%. So over half of patients, just over half, had a response to the therapy with a complete response rate of about 40%. And what's interesting here is, although we do have a PFS that's measured about in the 12-month range, or a little bit less actually, nonetheless, there's a plateau. And so for a significant amount of patients, so over 40% of patients have a long-term survival with this therapy. The second product, Tisa-cel, Axi-cel, sorry, or Yescarta, done by Gilead Kite, is also a CD19-directed CAR T-cell product. This one with a slightly twitchier CD28 co-stimulatory domain. Same sort of clinical trial, phase 1-2, about 100 patients. And same disease, diffuse large B-cell lymphoma, transformed follicular to diffuse large B-cell lymphoma. They added primary metastatic B-cell lymphoma to this mix. And again, these are patients that have had at least 2 previous lines of therapy. The median was 3. And many of them have had autologous stem cell transplant as their salvage. And again, we see results that are pretty much similar, with an overall response rate that's pretty impressive. And in patients that reach a complete response, here, yellow, there is a plateau. And so after a median follow-up of 27 months in this trial, about 40% of all patients remain in a complete response. So how does that compare to other therapies out there? And so the comparator really is a trial done by Mike Crump in Toronto called Scholar-1, where these are the same types of patients. These are patients that have relapsed after their primary therapy, have gotten salvage therapy, and then have relapsed again and waiting for further therapy. So prior to CAR T, the numbers were quite dismal. We're talking about a survival of only approximately 6.3 months. And very few of these patients actually get a long-term survival. So in comparison, CAR T looks far more interesting. So comparing these 3 products here, we see that they're similar. The difference being that they have slightly different co-stimulatory agents. There's a third product I didn't show you the data for called Lisa-cel or Breyanzi from BMS, with, again, in that same space, has a similar amount of response rate and similar long-term survivals. They're not terribly different. They do vary a little bit in terms of their efficacy, but not by very much. Again, we're talking complete response rate in the 40 to 50% range, and progression-free survivals that are different, and duration of responses that are similar, and duration of responses that are similar. Where they differ a little bit is in the toxicity, where the Axi-cel that has the slightly twitchier CD28 co-stimulatory domain has a little bit more higher grade cytokine release syndrome, and we'll talk about the toxicity. So a little bit more toxic, but maybe the trade-off is maybe a little bit more initial efficacy. So with that, these products, 2 of these were approved in Canada. The third is approved but not yet reimbursed. And as with all things in oncology, the current indication is going to change. So the current indication right now is at least 2 lines of previous therapy for diffuse large B-cell lymphoma or transformed follicular. And at the moment, they need to be, many of them will have undergone a stem cell transplant, an autologous stem cell transplant, unless they are chemoinsensitive or they failed it, which is their second line of therapy, or they're medically ineligible. They're too old, or they have some sort of comorbidity which precludes the use of autologous stem cell transplant. There's no age restriction currently, but they must have a good performance status and good organ function. So a good ECOG, a normal EF, and room air oxygenation, good kidney and function, and a good hematological reserve, so good counts, and at least, and this is important for manufacturing, an absolute lymphocyte count of at least 0.2, so you're able to have some sort of starting material to produce the product. So the indication will change, and sure enough, as we do in oncology very often, once we see success in later lines of therapy, we bring the product a little bit earlier, hoping that it'll have a broader range of applicability and as well longer outcomes or better outcomes. And so here in these 3 trials that were published relatively recently, this is for the same 3 products, CD19-directed CAR T cells, but instead of using them post-second line, this is post-first line. So there's a group of patients with diffuse large B-cell lymphoma, which when they relapse within 12 months, or they don't respond to their initial therapies, they're considered high risk, and their outcomes are dismal. Usually these patients will go on to some sort of salvage therapy. If they're young enough and well enough, they'll undergo autologous stem cell transplant. And this is the comparator for these phase 3 trials. So in these trials, the CAR T product, Axi-cel, shown here as an example, is compared to standard of care. So patients are randomized to receive either CAR T or standard of care chemotherapy at high dose, i.e. autologous stem cell transplant, should they be randomized to that arm. And the trials differ a little bit in terms of how they were constructed, but are basically the same. And some of them, one of them in particular, did not admit to any bridging therapy. So this is chemotherapy that you can give until the product is manufactured. And the other 2 did. And otherwise, the trials are similar. So the first trial was the Belinda trial, Tisa-cel. This is the Novartis product, where because of several reasons, one, their use of lymphodepleting chemotherapy, bendamustine, may have compromised, we found out since then, the utility of the graft or the efficacy of the graft. As well, their time to manufacture was extremely, extremely long. So patients lingered quite a long time, in fact, between randomization into this trial and reception of the actual product. And we think because of that, the outcomes were no better between stem cell transplant and the CAR T cell product in second line. Now, the other 2 trials were different, had different outcomes. So Liso-cel, the product that's not yet licensed here or reimbursed here, showed a very different PFS, progression-free survival, between CAR T and autologous stem cell transplant, so 10 months versus 2.3 months, and a plateau of the curve at around about 40%, 50% or so. So really a big difference between autologous transplant, which looks a bit dismal in this comparator. And finally, the ZUMA-7 trial. This is a trial that we participated here at the MUHC. Again, excellent differences between the standard of care autologous stem cell transplant versus the CAR T cell product, with progression-free survivals that are different, and again, a plateau of the curve. So, and I'm happy to say that as of a couple of weeks ago, there's a top line, more of a press release showing that it wasn't just the progression-free survival, but the overall survival, in fact, it was different between the two arms. So there's a clear advantage of giving CAR T and abandoning autologous stem cell transplant for patients with DLBCL that relapse within 12 months, so the high-risk early relapsers. So we'll move on to the other indication that's been approved by Health Canada, which is multiple myeloma, and obviously a disease that I'm close to. And in that disease, cure has never really been the target. We've never really seen cure with our therapies. We think more of myeloma as a chronic disease, which really benefits from successive treatments. So patients are treated once they have active disease, they go into remission, that remission lasts however long it lasts. They become symptomatic again, or their markers go up. We treat them again with something different and put them back in remission, keep the treatments going. These become chronically treated patients, which go on to a second relapse, a third relapse, until they're fully refractory to everything under the sun. And that's all we can offer them. And so this process typically can take anywhere from 7 to 10 years, we think, from the time of diagnosis. And but what truly happens to patients that are refractory to the available treatments? So the major classes of treatment in myeloma are the proteasome inhibitors. So bortezomib and carfilzomib are the main ones that we use routinely. The immunomodulators, as they're called, or cereblon binding agents, such as thalidomide, which we never use anymore, but lenalidomide and pomalidomide, and the monoclonal antibodies, the anti-CD38 monoclonal antibodies, such as daratumumab and isatuximab. So in patients in this study, and there's another one as well, that looked at about 300 patients that were refractory to our standard therapies. What we wanted to know here, what the authors wanted to know, is how they do with whatever you give them successively. So in patients that are triple refractory to these classes of therapy, whatever you give them, they end up having overall response rate of about 30% and progression-free survivals of 3.4 months. That includes clinical trials, that includes recycling other agents, and of course their survival is a bit dismal at 9 months after they're triple class refractory. So this represents an unmet need, and the unmet need obviously generates a lot of research, and one of them is a CAR T product. So unlike lymphoma, myeloma cells don't consistently express CD19, but they do express an antigen called B-cell maturation antigen. So this is expressed in mature B cells or plasma cells. And the first CAR T product to be devised is ide-cel here by Celgene BMS. And it's a product that, again, looks very similar to the CAR T product, with a 4-1BB co-stimulatory agent, with the binding portion to recognize BCMA in this case. In the trial, these were patients that are largely triple class refractory. So they're, again, that unmet need that I showed you in the previous slide. These are patients that received at least 6 lines of previous therapy on median, so heavily pretreated patients. The CAR T process is virtually identical to lymphoma. Patients are leukodepleted, it's manufactured. They're allowed bridging therapy. About 88% of patients required some sort of bridge, whatever investigators wanted to give until the T-cell product is ready, typically 3, 4 weeks, 5 weeks. And then they're infused. The overall response rate in this particular trial was 81%, with a complete response rate of about 40%, which is really impressive. If you compare it to what I showed you earlier from the comparative study, where the overall response rate with no matter what you choose is about 30%, and very few patients have a complete response with whatever it is you can devise. This is a departure. This is quite impressive for a single agent, one treatment. And unfortunately, it wasn't durable. So at the intended dose of 450 million cells, the patients that received this dose had a progression-free survival of about a year, 11 to 12 months, 11 months overall for the study. So a little bit disappointing that we're not seeing the plateau that we saw with lymphoma, but it's still a therapy. It's been approved by the FDA in 2021. It's been Health Canada approved in May of 2021. In the US, it's given quite frequently. They have supply issues. It's so popular. But in Canada, CADIF and INES, so the HTAs here, have given negative recommendations based on the trial concept and low comparator arm, and the fact that it is quite expensive for only 11 months of therapy. So it's not currently given and not currently reimbursed in Canada. So the second construct, or the second product, is called Cilta-cel, or now known as Carvykti by Janssen. And this differs from the previous product in that it has two antigen binding domains on its surface, but that bind the same B-cell maturation antigen, otherwise the same co-stimulatory agent. And in this particular trial, it was run almost identically to the previous one. These are patients that are triple-class refractory, that have seen on median 6 lines of previous therapy, and they're screened, enrolled, leukapheresed, manufactured, and lymphodepleted in very much the same manner. The outcome, however, is a little bit different. So we're not seeing 80% response rate. We're seeing 98% response rate with stringent complete responses in about 80% of patients. These are very, very deep responses. And to date, we estimate, we haven't reached median yet in these trials, but we estimate that the progression-free survival is going to be about 30 months or so. That's my estimation. We'll see what happens. Again, keep in mind that the real-world evidence, no matter what you give in comparison, provides a median progression-free survival of 3 months. And so this is pretty impressive. This is a huge difference. Again, used late in the treatment of myeloma. Ongoing trials in using them earlier in the disease if we can achieve even better results. So we don't yet have those data. So with that, it's been FDA and Health Canada approved. I'm happy to say that CADIF, last week, in fact, approved it for reimbursement. INES, their evaluation is still pending. It'll probably be this summer, I imagine. Don't know the dates yet. So these are the products right now that are either in the pipeline or approved and their indications. I didn't talk about all the indications, all the products, but there is an indication for acute lymphoblastic leukemia in younger adults. There is an indication for a CD19 product in acute lymphoblastic leukemia in all adults after relapse that is expected sometime this year. The approval, that is, and the reimbursement. Diffuse large B-cell lymphoma post 2 lines of therapy is given currently. And in second line, we're expecting that soon. Mantle cell lymphoma, I didn't talk about the data, but that's approved and reimbursed after 2 lines plus therapy. And finally, we're waiting for follicular lymphoma and myeloma very shortly. So expanding options. So what's the current state? So if I did a little clinicaltrial.gov search for all the active clinical trials in CAR T cells, and there appear to be well over 1,000 CAR T cells around the world, roughly split 60% hematological malignancies, 30% are solid cancers, and surprisingly, 10% of CAR T trials are for non-malignant diseases, such things as infectious diseases, HIV, autoimmune diseases such as lupus. There was a publication recently showing that a CD19 CAR T product showing a response in refractory lupus. So that becomes really interesting. And of course, this is the tip of the iceberg. This is the very beginning of what we're going to be doing with these exciting products. We're now currently using 2nd generation products, but there are further generations, 3rd and further generations of CAR T products where really we're limited by our imaginations. So things in which we can, throw in cytokines to help with the longevity of these cells or the efficacy of these cells. We can target dual things. We can have conditional CARs that will bind to one target but won't bind if there's a presence of another antigens, et cetera, et cetera, et cetera. We're now working with in vivo expansion. There are publications with allogeneic CAR T, so off the shelf. These are donor lymphocytes that are transformed. So really exciting. It's an exciting time for cellular research right now. So we're going to move on to the delivery and what it took to deliver this product at the MUHC. So we'll talk briefly about the toxicities, and of course we'll talk about the medical toxicities and those challenges. But the medical toxicities aren't the only toxicities that are associated with CAR T. There's what I call a logistical toxicity, as these are complicated to coordinate the manufacturing process. There's an administrative toxicity, so the toxicity that you come across in setting up the institution to deliver these products. There's a regulatory toxicity, so we have oversight with Health Canada and other regulatory bodies, and that proposes a particular challenge. And of course, there's a financial toxicity. So the acquisition of the products, at least the commercial products, are quite expensive. I can give you the list prices for most of the products. They range from $400,000 to $600,000 per patient. The actual negotiated price that the provinces pay are secret. I don't actually know. So we'll talk briefly about the medical toxicities. So the medical toxicities, there's an acute toxicity, which is the cytokine release syndrome and the neurotoxicity. There are delayed toxicity that we won't really talk about, or delayed toxicities that are associated with specific products when you squash down CD19, so cause long-term B-cell aplasia, so in terms of infection and immunity. So the cytokine release syndrome is a syndrome that we've been aware of for other mechanisms. But in terms of CAR T, it's directly engendered by the CAR T product binding to its target and then releasing cytokines and recruiting inflammatory cells and mediators to amplify that process, which also release more cytokine. The hallmark is fever, but can lead to tachycardia, hypotension, and a capillary leak syndrome, leading to hypoxia. It can lead downstream to organ dysfunction if it runs rampant, such as hepatitis and, again, respiratory failure. It also can lead to neurotoxicity. So neurotoxicity, if it occurs, usually follows a cytokine release syndrome, whereby the actual cytokines will damage or the blood-brain barrier, and the cytokines will penetrate into the brain. It's not the actual cells that penetrate. We know they're there. They've been seen there. We do CNS disease with CAR T and don't see much more neurotoxicity, but the actual cytokines themselves can lead to delirium, aphasia, seizures, et cetera, et cetera. So a particular issue in terms of management. So these are the key toxicities that I mentioned. They occur, cytokine release syndrome will typically occur 2 to 3 days after infusion. It can last up to a week. Neurotoxicity, as I've mentioned, if it happens after the cytokine release syndrome. So usually at day 4 to 6, and it can last up to 2 weeks, in fact. And again, these are engendered by specific cytokines. In particular, we pay attention to interleukin-6, which is a linchpin, and to some degree interleukin-1. And so we've developed, in looking at how to manage these patients, we developed an algorithm, if you want, or a process where we follow the trajectory of the patient from the manufacturing process to their admission to manage the toxicities post-infusion. So they're admitted to our unit on D10. We have a very specific mechanism of identifying these patients. They're identified by color-coded charts, and also on OASIS, there's a tab for risk factor, and it appears there, so anybody knows that they've received a CAR T product. We have specific pre-printed orders for just about every aspect of the process. There are also admission orders that are done frequently, and as well as monitoring by nursing and medical staff. We have a process of pre-admission notification. You may have seen this if you're a member of the ICU or neurology. We notify those that could come into contact with these patients that an infusion will occur on this day, and to expect, if it occurs, a cytokine release syndrome or neurotoxicity on the following days. And of course, we do our assessments. We try and prophylax as much as we can, and we keep our antidotes very close to the patients, our antidote being an anti-IL-6 receptor antagonist called tocilizumab. And of course, we have guidelines, and that's one of the first things we did, is we built specific guidelines that are really aimed at the house staff and also the medical staff, the nursing staff, that are found in an accessible location on the server. And so at any time of the day or night, these can be consulted. These are interactive. You can click on them, and I'll show you more of them later. You can click on various aspects and get information as to how, first, what CAR-T is, what the toxicities are, and how to manage them, specifically how to grade them and how to manage them. And so this is our management algorithm, which is interactive, where we sort of instruct what to do in different inpatient units, how to monitor. So this is our neurotoxicity monitoring forms. There's specific forms here, and then how to grade. This is an example of the cytokine release syndrome grading here, and management, exactly what to do step by step for each grade, and in what location, whether it's on D10 or once the patient has been transferred to the ICU, should they need ICU transfer for management of their toxicities. So this has proven to be extremely, extremely effective and helpful, especially with regards to house staff, who are really the front line. And I can't thank them enough. They're the front line in managing these patients in the middle of the night. So yes, they can and they do call us, but they have ready access to these tools. So how do we build the program? So, we approached this program with regulatory processes in mind. And we're accredited by the Foundation for the Accreditation of Cellular Therapy. This is an organism that really has oversight over cell therapy, such as stem cell therapy, so allogeneic and autologous stem cell therapy, but also CAR-T. And they put forth certain standards for which we get inspected. And so we had to be compliant with, and we had to adhere to these standards. And so these standards describe everything from the clinical units, the personnel, how they're trained, what their roles are, what their competencies are, what the maintenance of their education is like, whether we have a quality assurance management and plan. Obviously we do, and what that entails and what the structure of that plan is. They wanted to see all of our policies and procedures, how we select patients, how we evaluate them, and how we manage them, pre-infusion, post-infusion, any clinical research that's done, how we manage our data, so how we actually collect all that data, and do we participate in registries, which we do, and how we keep our records. Our standards in terms of the cell collection. So this is the cell separation unit on the 10th floor. The personnel, their training, their quality assurance, all of their SOPs, how they label. It's very important that you know that there is a chain of custody. This is a very unique situation in which the drug is manufactured from the patient. And so there has to be a clear chain of custody of every single step so that we don't manufacture the wrong product for a patient. They don't receive somebody else's cells, for example. So that chain of custody and its documentation becomes really paramount. And so the processing lab is involved in that chain of custody. It's involved in the storing and shipping. It's involved in some manipulations, minimal manipulations, and of course the storage and all of that documentation. I mean, everything from cleaning to the environment has to be regulated and is. And then finally, there is the contract, and that was a real big challenge in getting contracts set up. This is the first time the hospital itself has ever been part of the manufacturing process of a drug. And so it required a new way of looking at contracts and the role of a hospital. And so with these standards in mind, we didn't set out to build a program from scratch. We weren't that stupid. We realized that, there's a lot of common standards between the stem cell transplant program and immune effector cell program. About 70% of our content is actually overlapped. So we decided to really fuse together and join forces. So before we came about, this was the actual structure of stem cell transplant, from everything from the medical directors, the program director, to the director of the various aspects, from cell collection to laboratory to the clinical. And we're a combined program with the pediatric side as well. So we inserted ourselves right here, where we could share a lot of the managers, we could share some of the laboratory process and add our own, and that's what we'll talk about next. So that shared accreditation is what we really aimed for. We decided to go for a multiple point delivery, that being our D10 floor. We knew, and we still know, and we expect this indication to grow. So we expect the next users are going to be the medical oncologists for solid tumor. We're seeing signals in diseases like sarcoma. And so it's just a matter of time before effective products come on the market. And so we trained all of D10. We have common nurses, common educational standards. So we trained both the South and the North and Central pods. And so these multiple points of delivery were integral to our process. And so we couldn't do this without A, approval, B, funds. And so with really the approval of the institution and the help of the DPS at the time, Dr. Sidorovich, as well as Dr. Aprikian, who's the mission medical director, Christine Bouchard, who is our head of nursing for the medical mission in oncology, and myself set out to do some initial hiring, including a project manager, Selda Elmas, and a nurse specialist, which we dragged out of retirement, kicking and screaming. She was our D10 head nurse, who was heavily involved in FACT accreditation, came back to work with us. And with that crew, we then went to the MSSS for basically funds. We needed a program coordinator, a quality assurance officer, a laboratory quality assurance officer, admin assist, data archivists, and support for the technical aspects, as well as some physical needs that we had. And I'm happy to say we were quite successful in achieving all of this. And so next we worked with the administration, the pharmacy, again, the contracts, which we talked about, the hospital ethicist in building our consents, as well as the IT department. There's a lot of sharing and data sharing between the institution and the manufacturing site, which are in the US. So that created a lot of logistical issues in terms of data transfer and privacy assurance, and all that reflected in the consent process. So I think I'm going to skip all of this. I think we've more or less talked about the trajectory of the patients. But with that trajectory in mind, we built all of our SOPs and all of our forms. So we tried to protocolize as much as possible, everything from the indications, which are sent out to our partner institutions and used internally, to our SOPs. So I'm giving you one example. This is a 113-page example of a standard operating procedure, this one for the management of CRS, where every step of the management is detailed step by step, like painstakingly. And so with all of that built, and I won't show you the full amount of paperwork that we generated in doing this, but rest assured that it's quite huge. We opened an IUC core. This is the picture of us when we back last spring, essentially. And the core consists of, again, our nursing medical director, Christine Bouchard. I'm the medical director, the manager of the apheresis and inpatient unit, Julie Mathieu, the project manager, who's agreed to stick with us for the moment, our IEC coordinator, Janelle, our CAR-T coordinator, Giselle Wilson. We have 2 and a little bit more QA managers for the clinical program, as well as a quality assurance for lab, an admit assist, a data archivists, and our partner pharmacists. Our milestones were, well, we were accredited by Health Canada for cell therapy in 2019. Our last FACT accreditation was last spring. So keep in mind that we opened up this program during a pandemic and our huge FACT accreditation. So kudos to those that were involved. It was a tremendous amount of effort. We were site certified for Novartis in end of March 2022. And our first commercial patient was the 27th of June, just a few days before our official MSSS designation. We subsequently have become certified for Gilead Kite Yescarta and Tecartus, and we've just undergone our site certification for the myeloma product, cell-to-cell. So these are our stats. They're almost up to date. So far, we've received 24 referrals. It's been a little bit slow, probably because a lot of partner institutions are not yet aware that we're on the ground. It's going to take a little while before this grows, but we anticipated doing about 20 procedures in a year, and a year starting from our designation June, July till next June, July 2023. So we've been open for about 7-8 months, close to 9 months, in fact, sorry. And during this period, we've had 24 referrals. Not all referrals are obviously successful. Many of them fail for various reasons. So far, half of them have failed at various steps. We now have 12 completed collections, 8 infusions, 4 more upcoming, and more collections underway. Our median time from collection to infusion has been about 34 days. There's always delays. This happens. We aim for about 21, but 30 days is pretty much on average. Our ICU admission rate to date has been 20%, with a short length of admission. Tocilizumab use is 80%, and median hospitalization about 21 days. So far, all of our patients have had good outcomes. Again, we don't have many to put in that statistical box yet, but we will for sure. So I'll finish off by giving you 2 cases that are slightly different from each other. Among our first patients, the first, an 82-year-old woman with a history of left breast adenocarcinoma, who presented with a low-grade B-cell lymphoma in 1996. Officially diagnosed with follicular lymphoma that eventually transformed to DLBCL in 2017, for which she received R-CHOP and went into a complete remission. 2 years later, roughly, she's proven to have a recurrence of her DLBCL, we're fairly certain, and salvage chemotherapy is given. Salvage radiotherapy is given as well. She was then treated with polatuzumab, a monoclonal antibody, for 6 cycles, and referred to us for really refractory disease at this point. She's collected on May the 24th. She receives in June her conditioning chemotherapy. She's given her CAR-T product on the 27th of June. She develops a fever on day 2 with a ferritin that doesn't rise right away, but eventually does. By day 6, 4 days later, she still has a fever. She's given her first dose of tocilizumab as per protocol. The ferritin falls, and she's discharged on day 15 post-collection. This is her PET scan prior to the CAR-T product, where you can see all the various sites of her diffuse large B-cell lymphoma, with a complete or near complete remission This is just a kidney and the bladder. So all of the sites of disease are not apparent anymore. And unfortunately, she dies of COVID in January of 2023. So obviously, this doesn't protect you from COVID. If anything, you're at slightly higher risk. And it's an unfortunate outcome. Second patient is a 56-year-old, so a little bit younger, with small volume adenopathy in 2008, eventually gets diagnosed a DLBCL and given R-CHOP in 2021, has residual disease treated with RGDP, and his stem cells are collected for autologous stem cell transplant, but his disease progresses. So he's no longer considered a candidate for stem cell transplant. He's given a second salvage and referred to CAR T therapy. He's collected for T-cell in June of the 9th, and he's infused less than a month later, or a month later, I should say, post lymphodepleting chemotherapy. He develops a fever first day with some tachycardia, and he's treated symptomatically. Third day, the fever persists, more tachycardia, and a rising ferritin up to 1,360. He started on tocilizumab. He's transferred to the ICU because of the tachycardia and hypotension with BP of 89/60. He started on levofed. He's hypoxic. He has increased work of breathing, which I'll show you why in a second. And he's diagnosed with a grade 3 cytokine release syndrome, as well as confusion, an 8 on 10 ICE score. This is the score we use to grade our neurotoxicity, which gives him a grade of 1. And he's an AKI. Tocilizumab continues, and he's given IV dexamethasone at this point. And so ferritin rose up to 5,000. So really quite inflammatory. This is the ICE score, or at least one component of it, where we do handwriting. We essentially get them to write a sentence. And you can see at the onset of his neurotoxicity, he's unable to write a sentence. So a CT is done, and we can see a huge mass in his abdomen. So there was likely progression between the time his cells were collected to when his actual CAR T procedure occurred, and that contributed really to his work of breathing with this huge abdominal mass pushing up on his diaphragm. Nonetheless, by day 5-6, he's still in a grade 3 cytokine release syndrome. Still is confused, has received tocilizumab and dexamethasone, but he's noted to have a ferritin that's jumped up to 12,000, and a fibrinogen that's starting to trend downwards. Day 7, his LDH again keeps rising and peaks at 1,400. His ferritin is now 23,000, and he's in disseminated intravascular coagulation with a fibrinogen of 0.69 and a rising INR and schistocytes on the smear. So this is really the end product, if you like, of runaway inflammation. So he's met the criteria for a hemophagocytic lymphohistiocytosis and DIC, as well as some tumor lysis syndrome. He started on tumor lysis treatment, and he started on anakinra, which is an IL-1 receptor antagonist. And it's like a light switch. All of a sudden, his cytokine release syndrome resolves, his ferritin trends back down, and he's transferred to D10, and he's discharged home on day 53 after a lot of rehabilitation after his ICU stay. Is really what kept him back. And his disease, which was, again, mainly abdominal here, has completely disappeared. And he remains in a complete remission and disease-free. This was September, and his last update was just recently. So he's doing very well. And that concludes the presentation.
Michael, thank you so much, Dr. Sebag, for a wonderful presentation, and also obviously for all the work you've done launching this fabulous program. Really, congratulations. There's a lot of questions and comments. For the sake of time, I'm going to, we'll start with some of the questions, and then I'll ask you to finish them maybe in the chat. How do you foresee the scalability of this therapy? Will there be multiple commercial providers, and will turnaround times become even more of an issue?
So the scalability is interesting because, you know, as I said, there's a financial toxicity. So as the indications increase, I think it's not a discussion for us here, but for certainly health economists, as to what's affordable is going to become, and what the price should be is going to become an issue. Other centers are focusing on the academic CAR-Ts, and certainly we're going to move in that direction of having a facility to do these ourselves. So that can reduce the costs in a research sort of an environment. We're getting better and better at administering these products. In fact, one of them can be done as an outpatient. And of course, they're admitted that they have complications. So that aspect of scalability is interesting. In terms of the manufacturing times, there's a lot of research being done on in vivo CAR-T expansion, which is we don't wait 2 weeks to produce, to expand the cells. We actually introduce a very small number of cells along with some cytokine growth factors, proprietary processes, where they do the expansion in vivo once you inject them. So that cuts the manufacturing time to about 5 days. There's also off-the-shelf allogeneic CAR-Ts, which we keep in the freezer. That's being looked at as well.
Thank you, Michael. Other question, how do you see the role of autologous stem cell transplant following the first relapse, given the new data?
Yeah, so for DLBCL, it's pretty much written in stone. It's as good a data as we're ever going to get for this indication, and we're pretty much expecting a reimbursement. The actual NOC has been given for second line, so after first line, and I think that was the question, right? Or is it in first line? You're asking entirely.
I think it's second line after.
Yeah, so second line, it's happening.
Fantastic. Is this only for people in Montreal or people in the rural areas? Is this for basically for anyone?
Yeah, so that's really interesting. It's for anybody in the province of Quebec. We're open for anybody. Of course, there's some requirements, and I didn't show you some of these, but after we do the infusion and they leave the hospital, they're required to stay within 1 hour of the institution, of the hospital. Not any institution, our institution. And so we do partner up with the manufacturers, which have offered assistance programs in terms of housing and transport.
Fantastic. How do you see, what is the setup cost? Any comments about setup costs and annual operation costs for this kind of program?
Yeah, so I showed you some of the costs in terms of the personnel. So really the large part of the setup was really personnel. So paying for managers, paying for the coordinators, the data managers, and that is a recurrent budget that's given to us by the MSSS. And in terms of physical infrastructure, we're lucky that we have a factor-credited auto transplant program with that in place. So setting it up without a factor-credited program is a very different entity, where all of these processes have to be put in place.
Fantastic. How do you foresee improving the narrow window that lymphoma patients have in order to help the chance for patients of being offered the CAR-T treatment?
So a lot of that is early referral. It's a really good question. So we've really, a lot of our failures have been because these patients are referred too late. They come and they're too sick, their burdens of disease are too great. We don't think we're going to be able to get them through the collection and the subsequent wait time until the infusion. And so the best thing I can say is to refer them as early as possible.
Fantastic. Michael, for the sake of time, it's past 1 o'clock, so I will have to close the official rounds, but I do invite you to please, if you can stay a little bit longer to answer some of the questions and see all the lovely comments there are in the chat. You could perhaps do that and just perhaps answer in the chat, or if people want to stay on. Well, really, one lecture, comprehensive, encouraging results. So we're happy to see this obviously moving forward. And really congratulations, because it's this together. There's just always hurdles along the way. I think we all know that. And so to make this happen with all your SOPs and all the people involved and everything that you need on the ground to make this happen, kudos to you and your team. A tremendous amount of work and lots of patients, many patients will benefit from this. So we will have to close the rounds, but I invite you, Michael, to please read the chat, look at the comments and questions that are in the. Congratulations to you and your team once again.
Thank you.
Everyone for joining us this afternoon. Have a great afternoon.
Sasha, to answer your question, I don't know if you're still on. There's still people on. Okay, so the actual cost is a little bit difficult to ascertain, but essentially the ministry gives us a fixed cost per patient, not including the acquisition cost of the product. So, it's roughly about $50,000.
Okay, great. Well, yeah, that's great. Thank you so much for your wonderful.
You're welcome. And Ines, you asked about RA. I think you're still on, Ines?
Yes, I'm here waiting for your answer.
I don't know. That patient unfortunately died, but we did a patient who had, I believe, Sjogren's, and she was infused, I think, 3 weeks ago. So we'll see what happens.
I think that patient has cryoglobulin vasculitis, right?
Yes. Yeah. So we don't know. I mean, you'll follow her.
Yeah.
And I think Sasha's involved as well, if I'm not mistaken.
Evelyn is a physician.
It's Evelyn. You're right. It's Evelyn. Yeah, and we'll see what happens to their vasculitis.
Can you comment a little bit about on the samples that you get from the patients, and what are you biobanking, and what are
We're not biobanking anything at the moment. So we're just doing data management. There's no biobanking.
Do you have plans to do that?
We could. Yeah, we could. Definitely.
Thank you so much.
Okay, you're welcome. I gotta go back to clinic. Bye, everyone.